All right, welcome back. This is now Chapter 7, the control of microbial growth. Make sure you have thoroughly gone through Chapter 6, growth of microbes first. So in this chapter, we'll talk about the microbial death rate. In the Chapter 6, we talked about the growth curve and how to calculate generations and length of generation, which would be, the same idea, but of growth. And so today we'll be talking about death rate, and then different methods to either inhibit or kill microbial growth. We'll look at physical methods and chemical methods, and that's our summary. Okay, so before we get started, let's remind ourselves of some terminology. So sepsis means contamination, and asepsis, the A means without, right? So asepsis is the absence of, and we would really say significant contamination, because the only way to be actually without contamination would to be sterile, right? So asepsis doesn't mean sterile, but it means no significant contamination. And then we practice aseptic techniques in the lab to avoid contamination. And in that case, you're starting with sterile media and sterilized tools and everything. So you really are trying to keep everything as close to sterile as possible so that you can rely on your findings, right? So sterilization, as I mentioned, is removing all microbial life. And you're not actually removing the cells, but you're killing them. And they will degrade and fall apart. But it is possible to find intact cells, but they will be dead, right? You will have denatured all their proteins, and they will not be viable. If you were to transfer them into a nice environment, they would not grow because they are dead, right? And this is really difficult to do because it means you have to be able to kill endospores. And remember that endospores are incredibly resistant. You could boil them for like 20 hours before you would start to kill them. And no one's going to boil something for 20 hours, right? So that's why we use a special instrument, right? The autoclave is our best way to combine that heat, steam, with pressure. Allows you to obtain higher temperature and actually kill those endospores in a timely manner, like 15 minutes, and maybe about an hour for the entire beginning to end of the entire cycle. Okay, so commercial sterilization typically just means being able to kill Clostridium botulinum, so you are not going to give people botulism when they consume your canned goods. Disinfection, on the other hand, means to try to remove pathogens from a surface, for example, like your countertops, right? And you're not trying to sterilize it, but you're trying to remove the bulk of pathogens. And then antisepsis is trying to remove pathogens from living tissue. And so we call the products that you use on living tissue, we call them antiseptics, right? So sanitation, which is also a lot of times a term used in like industry, is just lowering the microbial counts on things like eating utensils, right? So restaurants will use this term, but it's not sterile, right? So a lot of people confuse sanitation and sterilization, not the same, right? You're just trying, the sanitation's just trying to prevent the spread of any illnesses So people feel safe using your products, eating at your restaurant, etc. Okay, biocide, side means killing, right? So biocide or germicide, viricide, viruses, right? So you'll see side at the end of all sorts of different microbial words, right? Different types of microbes. So if something just says germicide, it may only be referring to bacteria, but it also might be including all microbes. I don't really like the word germicide because it doesn't tell you specifically what it means, right? But it should mean all microbes, killing microbes. But a lot of people only really think about bacteria because they don't know about other types of organisms. So I'd just be careful that you clarify that it is actually all microbes and not just bacteria. So when you are talking about just bacteria, you're going to say like bacterio, and then you're going to give it an ending. If you're killing bacteria, it'd be bactericidal, right? If you're just inhibiting its growth, like putting it in the refrigerator or exposing it to an environment that's not really ideal, so it slows way down, then that would be stasis, right? To slow down, to inhibit, pause, static, right? So you're not actually killing it. Once the environment is nice and good again, it will take off and grow very quickly. And furthermore, during this inhibition, typically you're still getting slow growth, so over a long amount of time you'll still have a lot of microbes there. Okay, and you'll see that in your lab this week. Okay, so this slide's kind of ugly. Sorry for that. Okay, so the rate of microbial death, the objective describes the patterns of microbial death caused by treatments with microbial control agents. And we're going to go through a lot of different control agents, but first we want to talk about microbial death. Okay, so microbes die at a constant rate. It's proportional to the concentration of the microbes. The more microbes present, the more time you'll need to kill them. Okay, you don't just poof wipe them all out. Okay, and of course, the more of the killing agent that you use, so the higher the heat or the more concentrated the chemical, then you will speed up that rate of killing. So killing is dependent on how much of the microbe or how much of the organic material is present and how much of the killing agent is present, right? We have a graph we'll look at on the next page that demonstrates this, okay? And we're using a log, right? Just like we saw with microbial growth, we see microbial death is also logarithmic, okay? And so when a Lysol bottle, for example, says that it kills 99.9% of germs, that sounds like, wow, that's everything. It's killing everything. Well, let's take a look and see what we think about that. Also, that would be represented as 3 log. Okay, so 1 log, 90%, 2 log, 99%, 3 log, 99.9%. There's also a little graph in your book. This chapter is pretty good in your book. Make sure that you're using your book to help you understand the concepts. Okay, so this red line plotting the typical microbial death curve logarithmically, right? So using the log scale, and it shows you that it's a straight line, and that shows you that it's a constant rate. It's also a lot more useful because now you can get a better idea of how many microbes you're actually dealing with, right? So at each log, 1, 2, 3, 4, 5, 6, right? It also shows you time on the bottom, and then it shows you the actual arithmetic numbers of surviving cells. Okay, surviving cells, right? Because right now our goal is to kill. So when you use the arithmetic scale, once you get down, to even just 1 hour, it looks like you've killed the majority of the organisms. Once you get to 2 hours, you look like you've almost finished. In 3 hours, you look like you're done, like you've killed everything. But when you look at that on the logarithmic scale, you can see that indeed you have not killed everything, right? And in fact, not until you get out to 6 minutes on this scale can you actually say that you've killed everything. Excuse me. Okay, so the blue line's impractical. At 3 minutes, the population of 1,000 cells would only be 100th of the graphed distance between the 100,000, right, and the baseline. So this is a more technical way to explain what I said. It visually is deceiving, right? And you can't even tell how much you have at a certain point. So just like we saw with growth, it's impractical to use the arithmetic scale. We need to use the log scale. Okay, so I already talked about effectiveness of treatment, but let's talk about it a little more. So it depends on how many microbes are present. Also, is there other organic matter there to get in your way, right? How many of the microbial cells are there? What other material is there? Are there biofilms? That would be a huge question to ask. And temperature, right? What is the temperature that you will be performing the experiment at? How much time are you going to expose it to the treatment? And any other special microbial characteristics like an endospore or a capsule or a rapid efflux pump, right? If you're using a chemical, you're trying to use a chemical to kill, but it has a efflux pump and it just gets rid of the chemical right away, then you're not going to kill it, right? So you do need to have an idea of what you're working with. So let's go ahead and ask ourselves a question at this point. Do we understand this idea? So microbes die at a constant rate, and it depends on these factors, how fast you kill, right? So what would take longer to disinfect? 0.1 liter solution that contains 1 million bacterial cells, or a whole liter solution that contains 100,000 bacterial cells. Which do you think would take longer to reach full, let's say full, we shouldn't really use the word disinfection, I guess. Let's use the word for full sterilization, to kill them all, okay? Okay, so hopefully you thought about this question. And even though this has only 0.1 liter volume, it has 1 million cells. So it's going to take longer to kill 1 million cells than it is going to take to kill 100,000, even though you have a whole liter here. Now, some of you might be saying, wait, this is not a fair question because we don't know all the information. And you're right. This could change. If we're assuming that these liters are just water or some, buffer, whatever, even just growth media, that'd be okay. Then what I said would be correct. But if we start to introduce other factors that we talked about over here, then we could actually change this, right? So anyways, hopefully you got the idea of 1 million cells will take longer to kill all those than 100,000 cells, right? If the solutions are the same type of solution. If, on the other hand, in this liter, it's like sewage, where you have lots of organic material, then it would actually take longer to kill that in that case. Okay. So let's talk about different control agents. All right, so we can kill in different ways. We can attack different parts of the cell and have different kinds of reactions, right? So if we attack the membrane, we cause it to become more permeable, then it's leaky. It's going to leak out the inside contents. Also, with the membrane being weak, it's more susceptible to lysing open under that osmotic pressure. And so you would kill it, right? So attacking the membrane typically leads to killing of the cell. Remember the example that we've talked about a lot in class so far is alcohol doing this. Okay, we're going to see a lot of other substances, or at least a few. Okay, damage to proteins. We've talked about this a bit too, right? If you change the shape of the protein, it can no longer perform its function. And we've talked about the most popular way to denature proteins is just to expose them to high heat. We've also talked a little bit about using pH change, right? Extreme alkaline or extreme acidity can denature as well. And then we haven't really talked a whole lot about damage to nucleic acids. We started to in the last chapter, maybe it was chapter 5, where we started to talk about free radicals, right? And so oxidizing agents, which a free radical is going to oxidize, or it is, it's going to cause redox reactions. And so oxidizing agents are going to be damaging to nucleic acids. We can also use other non-oxidizing agents, but use like types of radiation to damage things like our DNA. Right, so nucleic acid means DNA or RNA, or any type of nucleic acid. Okay, so if you damage the genetic material, then you remove the ability to make new proteins. So it does not necessarily cause an immediate death like we see with the leaky cells, right? But it can just lead to a very slow. It really depends on how much damage you've done to the nucleic acids. So a lot of times these are static methods, but sometimes they are actually cytal. It depends on the method and the extent of damage. Okay, chemicals may bind to enzymes and be inhibitors of enzymes, right? They might bind to proteins and inhibit the protein's normal function. You might even be binding to DNA or RNA and stopping certain processes or greatly slow it down. Okay, so these are the main modes of action of control agents. So would a chemical microbial control agent that affects the plasma membrane also hurt humans? I want you to think about that. Maybe read this section in your book and see if you can answer it. All right, compare the effectiveness of moist heat, dry heat, the different methods of each. Think about filtration, think about temperature, pressure, right? Desiccation, drying out, and radiation. Okay, so we're going to talk about all these different physical control methods. So moist heat, you are killing by denaturing the protein. Okay, so you're going to break the different bonds, like hydrogen bonds are usually the first bonds you break. If you have a high enough temperature, then you'll even break the stronger covalent bonds that are happening between the side groups of the amino acids, right? The best sure way to kill everything and achieve sterilization is the autoclave, right? We talked about that. And this is steam under pressure. So steam, okay, you boil water boils at 100 degrees Celsius and it forms steam. So steam is about 100 degrees Celsius. And that's pretty good, right, at our regular atmospheric pressure that does kill most things. But if you want to ensure that you're killing endospores, you need to get it even hotter, okay? Unless you have, 24 hours to wait, um, which is not practical, and so that's not considered an actual method. So we need to increase the pressure so that we can actually get that steam to be even hotter, right? And it might sound a little confusing, but the pressure keeps it from, it takes more energy to overcome, to boil, to form steam, and then for that steam to stay together to actually cause that temperature increase. So, without going into too much physics, we'll just accept that when you put it under pressure, you can reach these higher temperatures. And so now you have, 121 degrees Celsius, for example. And you can kill everything in about, at 15 psi, pounds of pressure. It only takes about 15 minutes to kill everything, including endospores. So it's a very cool tool. And remember, it does take time for it to ramp up, and then it takes time for it to cool down and let that release the pressure gradually. So the entire cycle of the autoclave usually takes closer to an hour, but the actual length of time that it holds that heat and pressure only needs to be about 15 minutes. Okay, so boiling is pretty good for killing most pathogens. 10 to 20 minutes, you can be sure that besides endospores, you've killed all the pathogens. There could be some surviving organisms, but you're unlikely to get sick, which is why whenever there's a question of water safety, you would boil your water for 10 to 20 minutes. And so it's pretty good, okay? Tantalization is repeated boiling and cooling. And remember, way back we talked about freeze thaw. If you freeze something and defrost and freeze and defrost, you weaken the proteins in the membrane, and then you'll end up cracking open those cells. And that's a technique we use actually to get viruses out of the inside of the cells when we're trying to work with viruses. Tantalization is the same idea, but instead of freeze-thaw cycles, it's boiling and cooling cycles. And so this does the same thing, this repeated boiling and cooling back to back. The idea is that you are going to weaken the proteins and even possibly endospores. So the idea is to try to achieve sterilization, doing these rapid boiling and cooling cycles. Again, you cannot guarantee that you're getting everything, but you can ensure that it's better than just regular boiling. Then we have pasteurization, which we talked a lot about already as well, where you're not trying to kill everything, you're just trying to get the majority of pathogens, and you're also trying to lower the overall microbial load amount so that things don't spoil as quickly, right? So your milk lasts about 2 weeks in the fridge instead of just a day or a couple days, right? So you're not going all the way up to boiling. If you boil, you're likely to damage the texture or taste of whatever it is you're working with. So it's a lower temperature. The time can vary depending on the technique and temperature that's being used. You will look at the specifics of this in lab. Okay. So again, the goal is not to kill everything. It's the goal is to kill just enough to prevent illness and reduce spoilage. We use this. Remember, it originated on wine, and it's most popular these days for milk. We also use it for juice, and that's what you'll see in your simulation on Labsters for juice. Okay, so this is the autoclave I talked a lot about. I'm not going to go over all this, but the only thing I want to point out is, and they may have updated this in your book, I can't recall, but you should never heat a closed container. When you open it, it can like splash the really hot liquid into your face. So that's the only thing I wanted to remind you. And you should be practicing this even when you heat things up at home, not just an autoclave. Like, you really should not be heating closed containers unless they have a vent hole, or you're following a protocol to do it safely. All right. So pasteurization, as I mentioned, there are different techniques. It depends on the temperature and the amount of time. And so classic pasteurization was 63 degrees, so far below boiling, right? 100 degrees is boiling. And this, you have to do it for half an hour. Okay, so we call it low temperature, long time. Okay, LTLT. Then if you want to speed things up, you can get up to 72 degrees Celsius, and look at this, 15 seconds. Boy, is that faster. Much, much faster. It's harder to, of course, heat things up to a hotter temperature. And so we have an apparatus, which you'll see in your simulation, that does this. But yes, so you only have to get it up to 72 for a 15-second hold, and then you can start to cool it back down. And so this is called high temperature short time. Nice, straightforward, nice. Okay, then you have something that's called ultra high pasteurization. For some reason, it's really not pasteurization at this point because it's above 100 degrees. So 100 and up is going to be, a sterilization technique. So ultra high is going to 135, so this is under pressure, for 1 to 2 seconds. So super fast, crazy high temperature. This is a sterilization technique, okay? It's not really pasteurization. They got this idea from pasteurization. They've modified it to become a sterilization technique. Now, we've talked about the goal is not to kill everything, but just to reduce the amount of organisms. So you're reducing the risk of getting sick, and you're going to make your product last longer shelf life. So we have these checks, right, where samples are taken and processed to see, is our technique actually working? Sure, we're following these instructions, but how do we know it's actually working? So we need to test the sample and see how much bacteria is actually present. So you take a sample and you grow it on a plate and you count the colonies. For regular pasteurization, your goal is just to have less than a total of 1,000 colonies per milliliter. You typically would have less than that, but that's the maximum that you could allow and still consider the product to have been pasteurized. Okay. Sterilization now technically should have 0, right? But we can allow up to 10 CFU per mil, right? Colony forming units per milliliter. But really you should have less. Okay. So huge difference, right? 1,000 CFU per mil for pasteurization is what we call the threshold. Okay, so you'll see these threshold numbers again in lab, so definitely want to memorize that, add that on to the slide, take your notes, right? So pasteurization threshold, 1,000 CFU per mil. Sterilization threshold, 10 CFU per mil. That's the maximum that's allowed after treatment. Okay. All right, so I started talking to you about oxidation. One method to kill by oxidation is just dry heat, so like an oven. Of course, this takes a very long time, so at 170 degrees Celsius for 2 hours. Now, we typically cook our foods hotter than this, right? A lot of times things, when we're cooking something in our oven, we're usually going to like 350, right? Fahrenheit. So actually, we should check that. What's the conversion between 170 Celsius and what is that in Fahrenheit? Now you could simply just Google that, and it will come up for you. And then that'll actually help give you a much better perspective of when you cook something in the oven, how likely is it that you've killed the bacteria? Also, notice this is for 2 hours. The most I've ever baked something in the oven is probably 1 hour, and it's usually less, right? So, again, that just shows you're not killing everything when you cook. You're just killing enough so that you're not getting sick. And that's why you don't eat leftovers past that 3 to 4 days, right? Okay, so anyways, how do you kill by dry heat? You could use an oven. You could use a flame, right? We talked about flame sterilization of our inoculating loop, inoculating wire. So we use flame sterilization whenever possible. If it's waste, you could do incineration, or like I said, the hot air sterilization treatment. There you go. All right, so what is oxidation? It'll form free radicals or toxic oxygen species, which are highly reactive and are going to damage molecules like DNA, right? Large macromolecules. Why? Because it's going to constantly break bonds as that extra electron is being passed around. And as those bonds are broken and then reformed, they don't always reform in the same way. And so then you change the molecule. You can cause a lot of damage. Okay. Now, filtration is a method when something cannot handle high heat, right? So things like, a lot of times, like antibiotics cannot be exposed to heat. You'll break bonds and they won't work anymore. So we can do what we call membrane filtration, which are very, very tiny, tiny pores on a membrane. You filter it through. Everything that goes through the filter is called the filtrate, and that would be considered your sterile fluid. And then the membrane itself is going to have trapped any, for the most part, any bacteria and viruses that were present. And then, of course, you would put that in the biohazardous waste at that point. Okay, if you're talking about air, we want to make sure that, like, for instance, when we're working in the lab, we want to make sure that the air is being filtered to reduce the likelihood of becoming infected from working in the lab, right? And this has become very popular, so a lot of people actually have HEPA filters in their home air purifiers. I've seen them in vacuum cleaners, right? So they're becoming pretty popular because they're really good at filtering air. They remove pollen, dust, right? Pet dander. So they really help people with allergies. And they can also help remove microorganisms. With our pandemic, that became a super huge focus, right? And so it has a small pore size, not quite as small as our membrane filter, but pretty small. Okay. All right. So that's filtration. This is showing membrane filtration, where you, of course, in order to get the fluid to actually go through the membrane, which has very, very tiny holes, you need to apply a vacuum so that you're actually causing suction and pulling the fluid through. Remember, anything that's too large to fit through the pores is going to get trapped, left behind, and then the fluid that gets through is called the sterile filtrate, right? Okay, so physical methods, physical methods. Temperature. So we talked about high temperature. Now let's talk about low temperature. Okay, so low temperature can inhibit growth, slows everything down. All the molecules are moving slower. It takes longer to do anything, right? And so refrigeration is like hitting a pause button, or close to, right? So it's just slowing it all down. Deep freezing is an actual true pause button. Nothing is going to happen at the negative 80 degrees Celsius. And then you can defrost, warm it back up, and it will resume normal function. Lyophilization is freeze drying. So this is a special apparatus that will be able to maintain a very cold temperature, usually close to, I think, negative 35. And it is also removing a special pump that's removing water. And so it's freeze dried. It's frozen, but it's also been dehydrated. This is great for storage. And I think I talked to you about this in when we talked about growth, and then we talked about preserving cultures. And we like to preserve cultures by either deep freezing or freeze drying, right? And so that's another pause button, right? Inhibiting growth. Freeze thaw, on the other hand, I mentioned earlier, is a cycle of quickly freezing and quickly thawing, and doing that about 3 times to actually kill and burst open the cells. You make them very weak. Okay, so you're damaging the membrane and the proteins, and so that will kill things. We don't use freeze thaw very often as like we're trying to sterilize something, because we have other methods that are quicker. But we do use freeze thaw a lot in, like I said, when you're doing viral work, you grow the viruses in cells, right? Because they can only replicate inside of a cell, and then you need to get them out so you can work with them. So you freeze thaw. Okay, all right. High pressure, I also talked to you about how you can use high pressure to obtain higher heat, but high enough pressure itself will also damage the proteins in the cells directly as well. So you don't necessarily have to have the heat. If you have just high pressure, that can be good enough. But of course, combining it with high heat, high pressure and heat, then you know you're killing everything, right? If you're just doing high pressure, how do you know? You'd have to get to extremely high pressures to obtain the same result. Desiccation means to remove water, and if there's no water, then you're not going to have metabolism. So this is this can be a pause button, but it can also kill. So if you dry something out completely long enough and then it dies, other things, once you reintroduce water, they'll live again. So it just kind of depends. This is one reason why we dry our dishes before we put them away in our cupboards. If you put your dishes away and they're damp, you could have things growing. If you've ever had Tupperware smell weird when you pull it out of your cabinets, that's why. It wasn't dry. Well, sometimes if you have plastics, they've absorbed odors from your foods, right? But for sometimes people just are not drying, like in the lids, right? Areas where it's easy for water to get trapped. So you gotta dry things out if you don't want them to get funky, right? All right. Osmotic pressure is another way, and we use this a lot in food where we don't want to expose it to such high heat that we change its texture. And so we use osmotic pressure to preserve foods quite often. Remember that too little pressure is going to cause water to leave the cell, right? And too much osmotic pressure is going to cause water to rush into the cell. So too much osmotic pressure causes plasmolysis. Water leaves the cell membrane shrinks, pulls away from the wall. Too little osmotic pressure, water rushes into the cell and can burst it open. Okay. So radiation. There are different types of radiation. There's ionizing, non-ionizing, and microwaves. Ionizing radiation, such as X-rays, gamma rays, and electron beams, are super damaging. They're going to penetrate very well through organic material. It's going to cause lots of ionization, lots of free radicals, and it's going to be very damaging. Non-ionizing radiation, on the other hand, is like UV, and you know that UV is also damaging. We protect our skin when we go out in the sun so that the UV doesn't damage our DNA and cause skin cancer, right? So UV can damage DNA by causing, if you have two thymines next to each other in your DNA sequence, they can then bind together, and they shouldn't be binding together, right? The bonds shouldn't be between adjacent nitrogenous bases. The nitrogenous bases should be connected to the sugars, right? Not to each other. So this is causing bonds to form where they don't belong, and we call this dimerization because it's two stuck together, right? So this is how skin cancer can actually occur, is through UV exposure forming a lot of thymine dimers. A certain level our body can actually take care of. We have repair mechanisms, but if you get lots of damage, then you exceed the ability of the repair mechanisms. And we furthermore have more checks and balances to still try to get rid of any cancerous cells. But again, you can overwhelm your body, or it can just simply miss it. And if that then allows to continue to divide and multiply, then it can actually turn into a cancerous mass, which just one cell, even if it is technically a cancerous cell, is not considered someone has cancer. We all have at any given time we'll have a cancerous cell that our body will kill and get rid of. But it's when our body's no longer able to keep it under control and get rid of it, and it's starting to rapidly grow and multiply, that it's then actually cancer. Now, on another note, you can have, sometimes you do have cells that divide uncontrollably and form tumors that are not cancerous. And so, I don't want you guys to freak out if, if you do have a mass, until the doctor tells you that they're concerned that it's cancer, and until the test comes back that it is, you should never panic, because this is actually quite common, right? All right, moving on from that. Microwaves are poor. They do not kill microbes. They might kill a few just from the heat alone, but not from the microwave itself. Microwaves heat your food by causing them to vibrate quickly. The molecules of your food vibrate, and that produces a lot of heat to heat up your food, but it's not really killing any microbes. So don't ever try to rely on a microwave to make your food safe. All right, make sure you read through these questions. You should be able to answer most, if not all, of these just from lecture. If you get stuck, definitely use your textbook to help you, and then you can always follow up with me during office hours. Chemical methods. Okay, so we just talked about physical methods. Now we're going to talk about chemical methods. Maybe I'll split this into 2 videos. So let's go ahead and take a break. You can review your physical methods, take your notes, read your book, and then come back for part 2 for chemical methods. All right, good job, guys.